This is Lecture 9, Sensation and Perception, covering the visual system, and this corresponds to Chapter 9 from your textbook. Only reminder that Homework 9 is due at the usual time this Sunday, the 10th at 11 p.m. Your recommended reading this week is an article on inherited trauma. Inherited trauma is the observation that trauma can actually affect people's expression of genes, so it can express epigenetics and other physiological systems in the body, and some of those things can be passed on to progeny, to offspring, children. And so trauma can sort of affect generations past the person who experienced trauma. The article comes from Teen Vogue, which I think I need to acknowledge, but it's actually a really well-written article. So I highly recommend checking that out if it interests you.
So just some basic information about sensation and perception. Sensory neurons code stimuli from the outside, from the environment, into patterns of action potentials. And sensory information is then interpreted within the brain. And we hope that it's pretty close to the actual environment, but it's often like a mismatch from the actual environment. So we know through optical illusions that a lot of times what we think we see is not what we actually see. We know this from hallucinations as well. Hallucinations are associated with mental illness, but actually some form of hallucination, either visual or auditory, occurs in about 40% of the population. So 40% of the population has had some kind of false perception that would count as a minor hallucination at some point in their life. And then expectations for what we're going to be perceiving also affects the interpretation of sensation. So sensory receptors transduce or convert sensory stimuli into neural activity. And sensory receptors are tuned to specific stimuli. So the visual system is tuned to light. The sensory receptors in the eyes, which are photoreceptors, are tuned to pick up on light, right? They're not tuned to pick up on sound. In the auditory system, waves of air pressure, which is what sound is, creates vibrations that physically stimulate hair cells, which are nerve cells in the ear, and then this is transduced or converted into neural signals. In the somatosensory system, which is responsible for touch and balance, physical stimulation on the skin and within the body is transduced into neural signals. And then in the gustatory and olfactory systems, chemical molecules in food and air bind actually directly to receptors in the nose and tongue, and then create neural signals from there. Each animal has sensory receptors that have evolved for their needs. So as humans, our sensory receptors are tuned to our specific needs. Other animals have different sensory receptors and different sensory abilities that are tuned to their needs. So for example, bees can see in ultraviolet light. And this is an example. So bees can't see red, I think. What we perceive as red, bees do not perceive as red. So what we perceive as yellow, they actually perceive as red. But they can also see in this wavelength of light beyond violet, which is called ultraviolet, and this helps them find the tastiest part of the flowers and the tastiest flowers. A receptive field is a specific region of the world, which is like a set of stimuli in the world that stimulates a given receptor cell or neuron. So photoreceptor neurons, which pick up on light in the eye, code for specific spatial regions in the field of vision. And if you look at this image on the top right, these little circles, these yellow circles, each sort of represents what a different photoreceptor cell in the brain might code for. So each cell in the brain has like a specific spatial region in your sight that it codes for. And the receptor fields are actually quite a bit smaller than that, but this is just kind of a demonstration of that concept. When we move, the brain is actually performing rather complex functions to keep our perception of the outside world stable while there's so much changing information in our visual field. So movement, keeping a stable concept in the environment while moving, is actually rather complicated neural process. So maybe if you've ever been sitting on one train and another train leaves, this can probably happen in like a car or a bus as well. But you're sitting in a train and the train next to you leaves, starts moving forward, you might start feeling like you're moving backward. And this is just one example of the visual system kind of being tricked, and the complexity of making us, keeping us perceiving the world as it is, as opposed to incorrectly perceiving the world. Kind of interesting, birds don't have as complex of an ability to count for movement in their visual field, and this is one of the reasons they bob their head when they're walking. So if you see like pigeons walking and they bob their head back and forth, this is actually a way of keeping objects in their visual field stable while moving. So if they're walking, it would just be a constantly moving picture, and their tiny little bird brains can't account for that. So they move their head back and forth to just keep part of their visual field stable for a little bit. So they're more likely to see little pieces of food on the street. This is a fun illusion related to receptive fields. It's called the floating finger illusion. So you can hold your hands in front of your eyes with the tips of your pointer fingers touching. So maybe hold your hand like 6 inches from your eyes, maybe even less, maybe like 4. And then focus your eyes on the distance, not your fingers. And you should see a disconnected, like floating finger or floating sausage between your index fingers. And this is due to the overlapping receptive fields in the left and right retinas. So left and right eyes have different receptive fields, but they do overlap. And that's what this illusion is picking up on. And we'll talk a little bit more about receptive fields of the visual system later. Neural relays. So all sensory receptors connect to the neocortex through 3 or 4 other neurons. They kind of have to, it's not just axon to cortex. They have to connect to a couple other brain areas, and therefore a few other neurons, which are interneurons. If you remember, interneurons connect sensory neurons to motor neurons. Information is modified at each of these connections based on other inputs from the brain. So the areas that the sensory receptors are going through are also getting receptors, also getting information from the neocortex, for example. And also there's just existing features of these areas that change the information coming in from the connecting neuron. It's sort of like a relay race of information, allowing for the contextualization and more efficient processing of incoming sensory information. It's kind of like if you've ever played that game like Telephone, where people stand in a row and one says a word or says a sentence to the person next to them, who says it to the person next to them, and you have to say it quietly, so it gets changed slightly each time. And then it gets to the final person, and you compare what the first person and the final person said. And the brain is kind of like that, except in Telephone, the joke is kind of that it's so different, like it's been misinterpreted all along the way. But in the brain, this is actually a good process. It's efficient, and it kind of fits information into our pre-existing structures for information. Okay, sensory systems are processed somewhat independently in primary sensory areas. So there's an auditory sensory area, primary visual area. So they're processed from their sensory organs somewhat independently, and then combined in the cortex or integrated in higher level cortices. You might have heard of synesthesia, which is when one sensory system is stimulated, and this results in the direct experience of another sensory modality or another cognitive process. So there are people who have synesthesia. I think it's actually a pretty big percentage, like maybe 4% of the population has some form of synesthesia, where, for example, some people will perceive different, like a number as being a different color. So they'll say like 2 is purple and 5 is red, something like this. Or if you look up at the top, if 7 is yellow and 9 is blue, and you ask them, oh, this picture looks a little wrong actually. 5 plus 2 is yellow, and yellow and 7 will be kind of like synonymous with them. They might see yellow in their head when they think of 7. And this synesthesia is associated with creativity. It's also associated with better memory because if you're encoding information in multiple sensory modalities, in auditory and visual, there's a better chance that you're going to remember it. Ideesthesia is another phenomenon where some ideas evoke perception-like experiences. So an example of this is experimenters gave people these two shapes to the right, this kind of spiky shape and this blobby shape, and they asked which of the shapes is Kiki, which would you name Kiki, and which of these shapes would you name Booba. And about 95 to 85% of the people chose the same shape for Kiki and Booba. So decide for yourself which one you think is Kiki and which is Booba. And if you agree with about 95 to 98% of people, Kiki would be the spiky one, and Booba would be the more curvy one. And this effect is even seen, maybe to a slightly lesser degree, in children who are too young to read. So this is not just a matter of associating the shape of the word, the shape of the written word, with the shape. It's something more about this connection that we make between sound and images. Okay, just some general terminology. Perception is the subjective interpretation of sensation by the brain. So perception is subjective, and sensation is just the basic registration of physical stimuli that comes from the environment, from the sensory organs. So for example, tasting something, when you taste something, the sensation is just, sweet candy on your tongue, and the perception is, this candy is sweet. I'm experiencing this candy as sweet. And then topographical map is something we're going to talk about intermittently throughout the rest of the chapters. And this is spatially organized neural representation of the outside world. So this means that topographical map is when the cortex in the brain is kind of physically maps onto how things are in the real world. And this is an example. So in the sensory and the motor system, there's something called the homunculus, which means the sensory areas, the part of the cortex, of the sensory cortex, for example, that codes for lips is close to the nose, which is close to the eyes. And these are close on our physical face. They're also close on the cortex, for example. Okay, so moving on to section 2, functional anatomy of the visual system. The rest of this chapter is going to be about the visual system. So vision is the dominant sensory system for humans. It's incredibly complex. People, scientists, dedicate their entire careers to tiny portions, tiny phenomena within the visual system, and there are so many things we still don't understand. A large portion of the brain is dedicated to visual processing, so the entire occipital lobe, plus much more of the brain, as you'll come to learn in the following sections. And before we learn about vision, we want to make sure we're on the same page about some basics of like visible light and just how the eye works. So light entering the eye is of course the foundation of vision, light that's been altered by the physical environment. And light waves are all different lengths. These different lengths are perceived as different colors. So for example, red is a longer wavelength, and blue is a shorter wavelength, and green is like a medium type wavelength. Structure of the eye. There's the sclera, which is the white part of the eye, the cornea, which is the outer clear covering. The iris is the colored portion, the pupil is this hole that looks black, and this is what lets the light in. The iris opens and closes a little bit more to let more or less light through the hole, making the pupil look bigger and smaller. The lens focuses the light. The retina is the surface on the back of the eye that's catching the light, and the retina is covered in photoreceptor cells and retinal neurons. And then the fovea is this smaller area in the center of the retina, which has the densest distribution of color photoreceptors, and thus creates the most detailed vision. So the retina, light travels to the retina through the pupil. Again, photoreceptors are these specialized neurons in the retina that transduce the light into neural signals. And this is the rods and the cones that you probably remember. The rods are there in the center, these little pink guys, and the cones are these yellow guys. The retinal neurons are ganglion and bipolar cells. We're going to talk more about ganglion cells in this chapter. And then the ganglion and bipolar cells are kind of transparent. They allow light to come through so that photoreceptors directly pick up the light. And then photoreceptors kind of send some signals to the bipolar and ganglion cells. The fovea, again, is the central region of the retina, and receptive fields are at the center of the eye's visual field. So the periphery is this visual area that's outside of the fovea's receptive field. If you think about like peripheral vision, this is what's being picked up on this area outside of the fovea. Then there's this blind spot. So where our optic nerve, where all the axons of the ganglion cells leave the eye, there's a section of the retina that has no photoreceptors, so it's not picking up any light, which means that in our field of vision, there's actually an area that we don't perceive, and that's kind of covered up by our brain. It fills in that information. Our eyes are constantly moving around, even when we think they're not. They're making very small movements, so you don't typically notice your blind spot. But if you want proof of your blind spot, on the next slide you're going to see a cross and a dot, like this image to the right. Stand a couple feet away from your screen and cover your left eye, and then keep your right eye fixed on that cross. Move slowly back toward your screen, keeping your right eye fixed on that cross, and at some point notice in your periphery that that dot should disappear. And when that dot disappears, this is picking up on the blind spot in your receptive field. Okay, so if you'd like to do this, go ahead and pause the video and check out your blind spot. But I'm going to move on right now. Okay, so photoreceptors. As I mentioned before, light hits the photoreceptors, and this causes a chain of chemical reactions that affects the membrane potential of the neuron and releases neurotransmitter onto the connecting neurons. Rods are literally rod-shaped photoreceptors specializing in functioning in low light. So they're all sensitive to the same pigment, and they're good for night vision because they're very sensitive to small amounts of light. So I heard once that the human eye in complete darkness can perceive one single photon 1 mile away. So if there was total darkness and your vision was perfect, like maybe 18-year-old's vision, not a 60-year-old's, then you would be able to pick up one single photon with the human eye, just because we have these very, very sensitive rods. And then cones are literally cone-shaped photoreceptors that are specialized for color vision and high visual acuity, so seeing a lot of detail. And when we focus on something, this is in our fovea, so this is in our this is cones picking up on that. Cones don't work very well in low light, and this is one of the reasons that you don't see color when it starts to get dark. Everything looks like the same color. This is because our cones are not working. And then different types are sensitive to different pigments, 3 different pigments, and we'll talk about that a little bit later. So color blindness is something we're going to talk about later as well, but there's a gene for red cones on the X chromosome. So if you remember, women have the two X chromosomes, XX, so they can sometimes have 2 different types of cones, which two different types of red cones, different types inherited from different parents, which creates an ability in some women to see more hues of red compared to other women and men. Because of this X chromosome being recessive and being, because of this red cone gene being on the X chromosome and being recessive, red-green color blindness is more common in men. So this is the most common type of color blindness, and it's more common in men, unfortunately, than it is in women, because they don't have this it's recessive, so if a woman inherits it, she's most likely to inherit also one of the dominant red cone genes, which means she would still have red cones. Whereas for men, they wouldn't have that extra X to be dominant. Okay, so the retinal neurons, the first layer is bipolar cells, and then the first layer includes horizontal cells, which link the bipolar cells to the photoreceptors, and amacrine cells, which link bipolar cells to retinal ganglion cells, so this like little linkage process. In the second layer are the retinal ganglion cells, which we're going to talk more about later, or these are kind of the most important ones in this section, besides the photoreceptors. The axons of the retinal ganglion cells bundle together and form the optic nerve, which, if you remember, turns into the blind spot in the back of the retina. There are two types of retinal ganglion cells, the magnocellular, or M cells, and these are sensitive to movement, and they're found throughout the entire retina. And parvocellular cells, or P cells, are more sensitive to form and color, and those are found mostly in the fovea. Okay, so there's a number of different visual pathways, but after the visual information leaves the retina, these optic nerves from the retinal ganglion cells from each eye, they kind of partly cross and then partly join again in the optic chiasm. The optic chiasm is this little X place right in the bottom of the brain, and the way that the information joins is a little bit complicated, but the inner part, or the nasal side of the left eye, and the temporal part, or the outside of the right eye, the information from that kind of joins together at the optic chiasm and goes to the right side of the cortex, of the primary, of the right hemisphere, so the right primary visual cortex. So the information that you're processing on the left side, the left visual field, or the left side of the world, is actually processed contralateral, or on the other side of the brain. Left visual field is processed in the right visual cortex. And then the same thing happens on the right visual field, where the inside part of the right eye and the outside part of the left eye the information from those two eyes joins together at the optic chiasm, and it's sent through the left side of the visual cortex. So right visual field, the right side of the environment, is processed in the left side of the cortex. So after the optic chiasm, there are three routes to the occipital lobe, or the visual cortex. There are two main ones and one minor. I'm going to talk mostly about these two main pathways. I'm actually not going to talk about the minor one at all, just for the sake of simplicity, but I'll go into a little bit more detail about the two main systems. So the geniculostriate system includes all of the P ganglion axons. If you remember, we just talked about the P and the M ganglion axons, and some of the M ganglion axons. So this goes from the retina to this nucleus in the thalamus called the lateral geniculate nucleus, which is a really big player in the visual system. This goes to layer 4 of the primary visual cortex, the occipital lobe, and then after the occipital lobe, it's going to go to either the parietal or temporal lobe, and we'll talk about those pathways in a bit. The right and the left eyes send information separately to these areas, to both the LGN and the occipital lobe. So the second of the two most important routes to the visual cortex is the tectopulvinar system. This is comprised of the rest of the M ganglion cells. It goes from the retina to the superior colliculus, which is in the membrane, then to the pulvinar region of the thalamus, so still through the thalamus but a different part of the thalamus, to the visual cortices, and then again to the parietal and temporal lobes. So the parietal and temporal lobes are these, also called like the dorsal stream and the ventral stream. These are two pathways that go from that bring visual information to other parts of the brain. The dorsal stream is like the Howe stream. This goes from the visual cortex to the parietal lobe, and it guides movement related to visual information. So we move in response to visual information, and the dorsal stream is mediating that. And the ventral stream is the what stream, and that goes from the visual cortex to the temporal lobe. And this is mostly used for object identification, so naming like a mug, a pencil, seeing faces, recognizing people. That's what happens in the ventral or the what stream. Both of these streams have input from both the geniculostriate and the tectopulvinar system, and I know that's a little bit complicated, but just know that both of these systems have information from those two. Both of these streams have information from both of those systems. Okay, so a little bit more detail about the occipital cortex. Within the occipital cortex, there are at least 6 visual regions, V1 through V5. V1 is also known as the striate cortex because it's striped if you cut it open. Striate means striped. This is also known as the primary visual cortex, so that can be a bit confusing. These are often used interchangeably. So just know, like, I typically don't use striate cortex. I try, for the sake of simplicity, to say either V1 or primary visual cortex. Input from both M and P cells, those pathways in the geniculostriate system segregate into 3 areas in the V1. So there are 3 separate areas within the V1 that code for motion, color, and form separately. In the V2 to the V5, which is also known as the extrastriate cortex, which means like outside of the striate cortex, each of these visual regions has a different type of neuron and different layer structures, which is how they initially knew that they had different functions. Just the neurons looked different in those different areas, so they knew there was different functions. We're starting to learn a little bit more about what those different areas are for, but we're not totally sure about all of the functions. We do know that motion, color, and form all go to V2 after V1, so they still are segregated in V2, and then they separate after V2 into V3, V3A, V4, V5 afterwards. There are additional visual processing areas, of course, outside of the occipital lobe. This is the dorsal and the ventral streams, and there's specific regions within those streams that we do know about their functions. So there's the lateral intraparietal area in the Howe stream and the dorsal stream that's responsible for eye movements, or is involved at least in eye movements. The anterior intraparietal area is involved in the visual guidance of a grasping motion. If you're going to grasp a pencil to pick it up. And the parietal reach region, true to its name, is responsible for the visual guidance of a reaching motion. So if you reach out and grasp something, you're using both of those areas of the brain, both of those latter two. The information about dynamic form and color go to the temporal lobe through the what stream, the ventral stream. And 2 big areas here, or well-known areas here, are the fusiform face area, which codes for faces and other expert-level visual identification, and then the parahippocampal place area, which codes for landmarks like houses, buildings, mountains, things like this. So the fusiform face area is very interesting and associated with a number of interesting behavioral phenomena. As I mentioned before, it's also thought of as kind of like expert level visual identification area. So humans are experts at facial recognition. People who are experts in other recognition, like bird experts, for example, use the face area, this fusiform face area, when identifying birds, leading us to believe that this is also just an area that is used for expert visual identification of objects. Damage to the fusiform face area leads to a condition called prosopagnosia, where a person cannot identify faces, particularly familiar faces. So even the faces of close relatives cannot be identified. Some people even report that they can't identify themselves in pictures. They'll be looking at pictures of people and will say, "Who is this? Is this me?" in the picture. They won't be able to recognize their own face. Alternatively, there are individuals who are super recognizers, who are very good at recognizing faces and can remember approximately 80% of faces they have seen, compared to a population average of the rest of us schmucks who can only identify 20% of faces. So if you want to test your facial recognition ability, you can take part in this very short study where you use some of what I believe is pretty well validated facial recognition tasks. So Capgras delusion is another phenomenon that's associated with the face area. Capgras was a scientist who noticed this sort of theme of delusions in certain people, and these were not necessarily people with schizophrenia or delusional disorder. Sometimes this resulted from a brain injury or developmental issue. So Capgras delusion is the belief that a friend or a relative, someone close to you, has been replaced by an identical looking imposter, so that this individual might believe that their mother, for example, has been replaced by someone that looks exactly like their mother, but they just somehow know that this is not their mother. They don't have the same feeling when they look at this person as when they look at their mother. And it's generally believed to be a result of a disconnection between the brain areas associated with facial recognition and the amygdala, which, if you remember, is the part of the brain associated with emotion, so feeling emotion. And the result of this disconnection is a kind of a lack of that feeling of recognizing a familiar person. So when you see somebody that you know, you don't only recognize their face, you have a certain emotional experience. It might be rather minor, but you have some kind of emotional feeling when you see them that is also part of the recognition process. And without this, it might feel like that individual actually isn't the person that you think you recognize. If you're interested, there's a video of a patient with Capgras delusion, and then here's kind of a written example of what somebody with Capgras delusion experienced. This individual, I think, had more like long-term psychiatric issues, so it can be part of a schizophrenia syndrome, or it can be separate. Okay, so moving on to section 3, location in the visual world and how location is perceived. So visual fields, again, are regions of the world that are seen by the eyes, and information in the left visual field goes to the right hemisphere, and information in the right visual field goes to the left hemisphere. And the left visual field is perceived in part by both the left and the right eye. Right visual field, again, is perceived in part by the right and the left eye. And so these neural inputs are separated at the optic chiasm and sent to the contralateral side of the brain. In terms of coding in the retina, each ganglion cell codes for a small circular portion of the retina. So light hits the photoreceptors within the small circle, the ganglion cells connected with the photoreceptors in that small circle, and the connecting ganglion cells activated. So the location of an object in our visual fields is determined by where on the retina the ganglion cells are being activated. So once information about location on the visual field, so like the location of an object, gets to the LGN, it's organized into layers of the LGN, so different places in the visual field are coded in different layers of the LGN. And the LGN cells summarize the information coming in from the retinal ganglion cells. So many retinal ganglion cells might synapse on a single NGN cell, and it kind of summarizes all that information from there. And this happens again in a single cell in the primary visual cortex. Many LGN cells synapse and summarize their information onto a single cell in the visual cortex. The visual field is represented topographically on the primary visual cortex. The visual field is actually flipped over, so things in the bottom of your visual field, like the grass, will be coded on the upper part of the visual cortex. And the fovea is overrepresented, meaning that for the amount of space that the fovea takes up on the visual field, it actually takes up, has a relatively large amount of the neocortex is dedicated to things that were in the fovea, and this allows for greater detail for the information that was in the fovea as compared to the periphery. So the corpus callosum, just an observation of the corpus callosum, if you remember, this is this white matter tract, this kind of C-shaped structure in the middle of the brain that connects the left and right hemispheres, but notice that it doesn't connect the visual cortices. It only connects certain brain structures. So the frontal lobes have a lot of connections through the cerebral cortex. The left and right frontal lobes are communicating back and forth quite fluently, but the occipital lobe has very few connections to the right and the left through the corpus callosum, and this means that visual information from each hemisphere really isn't integrated between the hemispheres until after it leaves the occipital lobe. So there was a section on seeing shape from pages 305 to 309. It's really interesting how we see shape, but it's really dense information, and you've had to read a lot already for this week, so I'm not going to cover it. If it's interesting to you, by all means read it, but it's not, you won't be tested on it in any way. So instead we'll talk about seeing color, which starts on page 310 in your book. We likely evolved to see color so that we could spot fruit. If you remember that we were initially hunter-gatherers, and being able to spot red fruit was very important to our survival. The primary colors of light, or the basic primary colors, at least for the trichromatic theory, are red, blue, and green. So note that this differs from the primary colors in like paint, which is red, blue, and yellow. Trichromatic theory accounts for very early color processing very early in our visual system. And it's an explanation of color vision based on the coding of 3 primary colors of light. And this theory says that the color we see, any color we see, is a function of the relative number of red, blue, or green sensitive cones on our retina being activated. And it predicts that if someone's missing one type of cone, the number of colors they see is going to be restricted. So if they just don't have red cones, they're not going to see the colors that involve red. And this is true. This is the case in color blindness. So a little more detail about color blindness, that you have some context. Lacking red cones and lacking green cones produces similar results. As you can see down at the bottom, an individual who lacks red cones or green cones is going to see a similar picture as compared to somebody who has all 3 types of cones, as in the first picture. But red-green is the most common type of color blindness. Red or green cones that are lacking can be either functionally limited or completely lacking. So there's different sort of levels of color blindness. There's a rather, I was actually surprised to see this when I read a bit more about it, that color blindness has a prevalence up to 8% in men and 0.5% in females. So remember that it's X-linked, so unfortunately men are disproportionately affected by color blindness. If you if you wonder if you have color blindness, if you aren't sure, apparently some people find out relatively later in life. There's this test that you can take. And there's also these kind of cute videos of people using glasses that fix color blindness, seeing full color for the first time, that you can see on YouTube. As well, many animals, such as dogs and cats, lack the green cones. It helps them actually see camouflaged items because they can't see these because they don't pick up on green so much. And humans lacking green cones can actually see through color camouflage. Camouflage is not as effective for them, so sometimes the military uses humans who lack green cones for this reason. So the trichromatic model that we were just talking about explains the very first steps of color processing, and later processing of color can be explained by the opponent processes theory, which assumes that there are 3 pairs of opposing colors: black-white, red-green, and blue-yellow, and this is based on the behavior of retinal ganglion cells. So just a little introduction to this. On the next page, you can stare at the red-blue box for 30 seconds, and after you do that, focus on the white box next to it, and you should see a green-yellow after image on that white box. So if you want to try this, go ahead and pause your video and stare at the colored box for 30 seconds, and then stare at the white box, but I'm going to move on. Oh yeah, and this is another one. This is a famous individual that you might not be able to recognize based on this negative image, but for similar reasons as the box before, if you stare at her image for 30 seconds as well, and then stare at the white box, you should be able to recognize her. Okay, so back to the opponent processes theory. Retinal ganglion cells respond differently to stimuli in the center of the receptive field as compared to the outer portion, and it's actually quite an opposite reaction that they have in the center versus the outer portion. So their kind of baseline response is this like intermittent firing, and this is a response to white light. In the case of a red-green sensitive ganglion cell, the cell, I'm kind of anthropomorphizing or personifying the cell here to explain it, but it sort of like prefers red in its center. And since green is the opposite of red, it likes green in the outer circle, so it kind of wants the opposite. The presence of green on the outer circle is almost like having red in the middle. It kind of gives the same strong response. When there's green light in the middle circle, it doesn't like this, and it gives a weak response, but it's still a weak response that's a little bit less than its baseline response. When there's red in the center and green on the outside, this is its absolute favorite, and it's a very, very strong response. There's strong contrast between these two colors. And then when there's green in the center and red on the outside, it does not like this, and there's no response. The cell sort of expresses its preference through the frequency of action potentials. So more action potentials for the color combinations that it prefers, and this is how these retinal ganglion cells code for what color they are seeing in the receptive field. So color constancy is this other function of color in our brain where we see colors when there are multiple colors in a system and we already know what they are, or in our visual field and we already know what they are. They will look similar even when there are changes in luminance in the environment. And the V4 in the cortex, so the visual cortex, is likely responsible for this. So a while ago, maybe last year, there was this image of this dress, the middle image here, that was circulating, and people couldn't seem to decide whether it was a black and blue dress or whether it was a gold and white dress. And the reason that we were not able to distinguish this is because we didn't have context and weren't able to establish what the colors were and then maintain this even in different luminance. So it was kind of poor luminance that made it unclear what these colors were. And in case you're wondering, I think somebody did some research and figured out that the dress was actually blue and black, the blue and black one. And then up in the corner here, this is another luminance illusion. And so typically people would perceive the bar in the middle of the box to be starting out light colored and getting darker, when in reality the bar is all the same color. So to test this, you can just cover up the background and notice that the bar is actually all the same color of gray, but because the context is changing behind this bar, it makes it look like the bar of the actual color is changing. I'm sorry, the shade of the actual bar is changing. Okay, so section 9.5, the visual system in action. Injury to the visual system can show some pretty interesting effects. So injury to the pathway on the way to the visual cortex. If the retina or optic nerve is destroyed, so if something early in the visual system is destroyed, the sight is lost in that corresponding eye, and the sight is lost, of course, ipsilateral, because if it's before the optic chiasm. Lesions to the primary visual cortex can cause scotomas or blind spots in the visual field, and that would be in the ipsilateral visual field. So notice in this image, in the picture, the damage is to the left visual cortex, and the scotoma is in the right visual cortex. And the degree of scotoma is dependent on the degree of injury. So a bigger injury causes bigger blind spot in that visual field. Blind sight is a condition in which someone who has severe damage to both sides of the V1 might report having no sight at all. So they would say, they would report being completely blind and genuinely feel completely blind, but there may still be some visual information being processed that is not available to the conscious awareness. So someone with blind sight may be able to guess details of an object in front of them with above chance accuracy. So if you say, am I holding a picture of a dog or a cat in front of you, somebody with blind sight would be able to they would feel like guessing to them, but they would be accurate above chance. So there would be some kind of visual processing happening. Similarly, if someone throws a ball at them, they might move their hand to catch it, sort of involuntarily. So if you throw it sort of at them like it's going to hit them, they might automatically move to catch it. They might not catch it, so the visual system is not good, but they would react to that visual information. So injury to the what, or ventral pathway, which goes through the temporal lobe, can result in a number of different things. One of them is visual form agnosia, or the inability to recognize objects. So agnosia in general means like lack of knowledge. A means without, gnosia means knowledge. Visual form, agnosia means the inability to recognize visual form. And there's some YouTube videos you can watch of patients who have visual form agnosia, but on the right is also an example of what somebody with visual form agnosia might draw in trying to replicate the drawing on the left. So really just not able to process visual form very well at all. Achromatopsia is the inability to distinguish color, and this is damage to the V4 region, so kind of the extrastriate cortex. Akinetopsia is the inability to perceive an object that's in motion. So individuals with akinetopsia can still see stationary objects. This is caused by damage to the V5 region. They can still see stationary objects, but once the object starts moving, they literally just can't see it. It's like the object disappeared. When the object is still again, they can see it again. And so patients with this damage have described that it's difficult to, for example, like pour water into a cup, because it just seems like the water is stationary and they can't see the glass filling up, so they might fill it to full. Akinetopsia can actually be temporarily induced by TMS, so transcranial magnetic stimulation. So you can put that coil, that figure 8 shaped coil, over somebody's V5 cortex and cause a temporary inability to perceive an object in motion. Injury to the how, or dorsal pathway, can result in orientation agnosia, which is this inability to recognize or inability to orient objects correctly. So there's a task where an individual has to rotate a card such that you can put it into a slot at the correct orientation. And you can see these circles with the lines through them down here at the bottom is like a control patient. The slot in this case was straight up and down in that task. So control patient, for the most part, was always able to insert the card at the correct orientation into the slot, whereas the patient, if they just had to match the orientation, like if they didn't sort of have feedback from the slot, they were almost completely at chance. They were unable to move this card in the correct orientation into the slot. In hemispatial neglect, an individual, this is damage again to the dorsal pathway, and in this case the individual doesn't process or neglects one side of the visual field due to this lesion in the contralateral dorsal stream. So if they have a lesion in the right dorsal stream, they're going to neglect the left side of their visual field. And this clock drawing is an example of a drawing you might see if you ask somebody with hemispatial neglect to replicate a clock. So you show them a picture of a clock and ask them to draw this. They know that there's 1 to 12, but they literally just don't process, or it really seems like they almost just ignore the left half, the left visual field, or the left half of the picture altogether. There's another example of a patient with hemispatial neglect who was asked to recall, I think she lived in Rome, and she was asked to recall like a famous square in Rome and describe everything on this famous square. There were many sculptures and things like this, and she closed her eyes and described everything she could sort of visualize in the square, and when they sort of drew it out, they realized she was only describing things that were on the right side, the side that didn't have hemispatial neglect. So she just didn't even have visual memory of the left side of her visual field. Okay, and this is just a quick summary that the ventral visual pathways are kind of associated with recognition, and the parietal visual pathways, or this dorsal visual pathway, is associated with the how, or like grasping objects, using the correct orientation of your hand, etc., things like this. Okay, and I believe that's it.